So let's just kick out for the first talk given by Vivek Gopalakrishnan. He's an undergrad student researcher in the lab, working with Professor Joshua Vogestin in Department of Biomedical Engineer. And the topic of his talk today is statistical methods for multiscale comparative connectomes. So yeah, so Vivek, maybe you can take over the stage and start your presentation.
Sure, thank you very much. Yeah, as Anqi said, I'm an undergraduate researcher in the Department of Biomedical Engineering at Johns Hopkins. Today, what I'm going to be talking about is methods for comparing connectomes across subjects. And the specific application that I'll be talking about today is for mouse models of autism. So just to briefly talk about what the connectome is, the connectome is this figure that you see here over on the right. It's this map of functional and structural connections within the brain. And the way that I think about it is that the connectome is very similar to the genome in a lot of ways. Just as measuring and enumerating the genome allows you to really understand the basis for genetic disorders, I think the promise of connectomics is the ability to understand neurological disorders by reconstructing these maps of the brain. But there are challenges, and I think the challenges can be best understood if you compare the connectome and the genome. So for one, the connectome is not an absolute unit. There are multiple sort of scales that you can view the connectome at. And in addition, the connectome also evolves over time. If you measure the connectome at 2 different time points for the same individual, you might end up seeing different things. And so the variability in the connectome really makes analysis very challenging. And so what this talk talks about today is some methods for doing principled statistical analysis of the connectome motivated by recent advances in graph theory. So just to highlight some of the existing methods that exist for comparing connectomes, I've got 2 different approaches that are very popular in the literature. In the first approach on the left, what researchers sometimes do is they will find a bunch of different structures within the brain, enumerate various properties about that structure, like for example, the volume of that structure, and then use that to compare connectomes from different people. So very simply, you could say, how does the size of the isocortex differ across a bunch of different subjects? The problem with this approach is that it really doesn't consider the connective structure of the connectome. It treats each part of the connectome as like a separate entity, and it doesn't consider connections between them. The second approach is to use some sort of graph metrics. These are functions that will measure properties about the graphs using just some sort of formula, and they're very, very popular to use. And while they give you some sort of measure of connections between different parts of the brain, they can be sometimes misleading. And this is a really great illustration of this. It's an analogy to Anscombe's quartet, if you've seen that, where like you have 4 different data sets that all give the same linear regression. Well, these are 4 different graphs that all give you the same set of graph features. And so the point behind this is that just if you're using graph features, you might not capture the true structure of the connectome well enough. And so that really motivates this project because we need better methods for comparing connectomes. So this is basically the abstract slide of this talk. This is really the heart of what my work focuses on. Just to give a quick overview, we start with a bunch of connectomes from different subjects that are all defined on the same vertex set. So that means that all the same regions of the brain are similar across all these connectomes. Then we go to the middle panel where we fit these connectomes to different random graph models that can tell you interesting things about the structure of a connectome at different levels of the graph. And then finally, what pops out at the very end is rankings of the most important edges, vertices, and communities within the connectomes. So broadly speaking, these are like just structures within the connectomes. And what these algorithms allow you to identify are the parts of the connectome that are most different across a bunch of different subjects. So just to quickly talk about the data that we're looking at in these connectomes. These connectomes come from 4 different strains of mice, with the BTBR mice at the top left being a mouse model of autism. And so these data were collected by some collaborators of ours over at Duke University, and they provide a really high fidelity data set for verifying our algorithms. I won't go too much into the mathematical detail in this overview. I'm just going to talk briefly about what these algorithms allow us to understand. And if anyone is curious about the algorithms, I'm happy to go into more depth in the Q&A section. So this is one of my favorite figures. It shows you really the heterogeneity in brain structures that we're able to identify with these algorithms. So I think the middle row is my personal favorite. If you see the panel that's outlined in blue, this is a rendering of the corpus callosum, which is one of the main structures within this mouse brain. What you're seeing is that in the one that's outlined in blue, that's the corpus callosum for the autism mouse model. And you'll see that there's very few connections between the left hemisphere and the right hemisphere. Whereas if you compare all the other corpus callosums from all the other different mice, so that's all the middle row, you'll see that there's many more cross-hemispheric connections. So what these algorithms really allow you to do is find the components of the connectome that are really different across your subjects, things that help you identify the subjects across these connectomes. And then in addition to looking at various components, you can also look at the brains as a whole. So what's going on in this figure is you've got a bunch of different dots on these two scatter plots. Each dot represents a mouse brain. And what we've done is some mathematical transformations that'll take the whole structure of the brain and embed it within 2 dimensions. And what we can see is that these embeddings are separable, meaning that you can identify the genotype of the mouse by the way that the graphs cluster. And so this is a really, really cool result because what it tells you is that you can separate different brains based on various phenotypes or genotypes of the brain. And so in total, this work is kind of an example, a case study on a specific data set, but our hope is that these algorithms will be used by tons of other researchers to do all sorts of cool connectomics analysis beyond just mouse brains. And just to wrap it up, I want to thank all my co-authors and collaborators. As well, all of the code that was used to do this analysis is available through the 2 packages listed here. And then I also finally want to thank my parents. Yeah, happy to discuss any questions.
Thanks, Vivek. It's really interesting. So this is something I forgot to mention. This is an interactive session, so people are welcome to ask questions, discuss even detailed or technical questions if they want, because we have some time left before the end of this talk. And just there's no stupid question. Just ask question, whatever you feel like confusing or not following enough when during the talk. So okay, so here I see one question pop up in Q&A from Aldana in the audience. He or she asked that, how did you construct the connectomes? That's actually also something I was thinking while listening to the talk. Can you go a little bit deeper into the construction of the connectome?
Absolutely,
a little bit?
Yeah. So the connectomes were imaged and reconstructed by our collaborators over at Duke. So what they did is they had 8 mice per each of these genotypes. They imaged them with ultra high resolution MRIs, and then they did tractography to reconstruct the connectomes. Our lab really focuses on the data science side of things, so we didn't have to do any of the imaging stuff. We just got the graphs and we got to do all the analysis.
Cool. Maybe a bit follow up on that. So yeah, because I've been working with some fMRI data before. So I know people have spent a lot of time trying to even coming up with this connectome from the fMRI, which is a big thing. But after getting this connectome, it is a really interesting problem how you match or compare a connectome across brains to learn something that can be shared or not shared across the animals or subjects. For this one, I think a quick question for the connectome is, how many connectome or how many nodes you have? Are they spanned over the whole brain? And when you compare connectomes or graphs, what kind of thing you use to match them together? Like the strength. I mean, because nodes, the distance between nodes are not necessarily the same across animals, and also some might have additional connectomes or links between nodes that don't really show up in other animals. So can you go a little bit more detail on that, how to compare that?
Absolutely. So the way that our collaborators reconstructed these connectomes is that every mouse was matched to the same atlas. So that means that every mouse has the same set of nodes. All the ROIs are the exact same. And in total, there were 166 ROIs, and there were 166 per hemisphere. So a total of 332.
I see. Can you be a little bit more specific about what algorithm or what type of algorithm you use to do the graph matching thing? I'm just curious to learn about more.
Yeah, the graphs they're already matched. It's one of the great things about this data set. We already know the labels of every ROI, and we know the tractography between each ROI. So we already have the matching. We already have correspondence of the vertices across all the connectomes before we even start the analysis.
So what's the most like non-trivial statistical problem you try to solve? You probably mentioned that in the talk. I kind of probably missed that, but just trying to understand if the graph is perfect matched on a template with the strengths between each nodes, can you just compare pair by pair, or is there any other statistical difficulties among that?
Sure, I'll hop a little bit into the statistics of this. So this is I think one of the cool answers to your question is when we talk about how we find which vertices are most different across all the different mice. This graph shows you 3 different approaches. The one in orange is the one that we use, and I can quickly break down how that one works. Basically, it's a method called graph embedding. So what that means is that you take this graph object, like all these connections between it, and you take it from a graph object and embed it in Euclidean space. And what happens is essentially every vertex is then associated with a vector. Every vertex can be described as a numerical vector that corresponds to some properties about the graph. And this is more to do with like the statistical side of things. But the comparison, figuring out exactly how to compare these vertices was the biggest challenge.
So that mostly happened in the embedded space.
Yes.
I see. Cool. Thanks for that question. One more question from the audience from Marco. The question is, we can use your program in auto-connected graph or necessary connectomes. He actually mentioned that it's weighted connected graph. Basically, can we use this program or this way of graph matching in other weighted connected graphs or connectomes?
Absolutely, yeah. The only requirement for these algorithms is that all the graphs need to be defined on the same vertex set. That's the one requirement here, because that's what we see in this first panel over on the left. All the graphs are defined on the same vertex set, so comparing vertices or different structures makes sense. If you had graphs that are on different vertex sets, you could take like a largest shared component across all the graphs, some union of all the graphs, and then do all the downstream analysis like that.
Okay, cool. Hope that answered Marco's question. Thanks again for Vivek's great talk and the answers to all these questions. Let's have